Good morning and good afternoon, everyone. My name is Mahalea Marshall, and I'm with the IQ Consortium Secretariat. On behalf of the IQ Consortium, I'd like to thank you for joining this webinar entitled Metabolism and Excretion of Therapeutic Peptides, Current Industry Practices, Perspectives, and Recommendations. This webinar is presented by the IQ Peptide ADME Working Group and is sponsored by the IQ Translational and ADME Sciences Leadership Group. Today's speakers are Michelle He from Eli Lilly and Sean Xiaojun Xu from Takeda. I'd now like to introduce today's moderator, Jesper Hammersgaard Christensen from Novo Nordisk. Dr. Jesper Christensen has over 10 years of pharmaceutical industry experience within the development DMPK area. He is an ADME specialist at Novo Nordisk in Denmark. His interest is primarily within peptides and oligonucleotides. He is co-organizer of the European DG peptide and oligonucleotide ADME workshops. With that, I'll go ahead and turn things over to Dr. Christensen.
Thanks, Maya. Thanks for the kind introduction. So excited to be here today, where we're talking about ADME of peptides, so what's not to like. So this working group is one of, if not the reason why Novo Nordisk joined the IQ Consortium. So looking forward to this great presentation and hopefully a good discussion afterwards. Besides this webinar, the working group will present a poster at the ISSX here in Boston starting on Sunday. So a little introduction to the topic. Therapeutic peptides, which we abbreviate to TPEPs, differ from small molecules and large proteins. Furthermore, submodalities of TPEPs have different structures and carry different ADME properties. There is no distinct regulatory guidance on conducting ADME studies for TPEPs, what, how, and when. Therefore, the Peptide ADME Working Group sponsored by the TAL of the IQ Consortium was formed with a goal to develop a white paper focusing on metabolism and excretion studies to support discovery and development of TPEPs. In this webinar, we will present key learnings from our IQ survey, industry survey, and FDA EMA submission documents of TPEPs approved between 2011 and 2022. We will touch upon TPEP's considerations for metabolizing safety testing, MIST, and not least, recommendations on conducting TPEP metabolism and excretion studies for regulatory filing. The presenters will be the co-chairs of the working group. Dr. Michelle is an executive director at Eli Lilly and Company with over 28 years of DMPK experience in drug discovery and development of small and large molecules, peptides, and novel modalities, including ADCs, and siRNAs. Michelle is serving at the editorial advisory board for the scientific journal Drug Metabolism and Bioanalysis. She has a strong record of publications with numerous presentations at national and international scientific meetings. Dr. Sean Xu is a scientific director at Takeda Pharmaceutical, where he's the head of Global Biotransformation and Isotope Chemistry Group. He has extensive experience in biotransformation and ADME to support discovery and development of small molecules, peptides, and large molecules. Sean has published around 30 articles in peer-reviewed journals. After the presentation, we will go in the Q&A, where also another group member, Constance Hillendorf from AstraZeneca, will be joining. So I will give the word over to you, Michelle.
All right, thanks. Let me share my slide. So I'm going to start with the introduction of the Peptide ADME Working Group. And so currently we have 14 working group members from different companies who are actively working on the peptide space. And Jesper is our sponsor after Winchi retired. So both of them have provided great, the guidance and the directions for the group. And so Sean and I are serving as the working group chairs. And so we were presenting all the work we did in the past year and a half. So also Constance will join us for the Q&A. To move to the next slide. So why Peptide ADME Working Group? As you know, peptides are different from the small molecules and the large proteins. and therefore it deserves the special attention or special consideration for the ADME characterizations. The expectation of following a small molecule like paradigm may result in generating a lot of ADME data, which may not be relevant in some cases. So it is critical to develop an industry consensus on generating scientifically relevant ADME data to support the development of peptide drugs. So our mission is to develop a guidance and the best practice for peptide metabolism and excretion studies. So the little more detail on the scope of the working group. So in the past year and a half, so we have been collecting study information from the IQ members, companies, regarding the metabolism excretion studies at a different stage of the development. And we also collect the regulatory interaction experience from the IQ members during the survey. And also we reviewed all the data packages from the regulatory and in the past decades. And also we defined the fit for purpose practice and the methodologies for the metabolite profiling and from the scientific perspective. And also we have discussed and make a recommendation that what the metabolism excretion studies will be required for the regulatory submission for different types of peptides. And just a note here that since our working group is really focusing on the metabolism and the excretion of the peptides, so the absorption or PK and distribution and the drug-drug interaction of the therapeutic peptides are not in the scope of this presentation. Let's move on to the IQ survey. The working group, we launched the IQ member survey in last year, and we mainly focus on the linear and the cyclic therapeutic peptides we call TPEPs. And so I will talk about this more detail in the next slide. So the peptides are divided into 3 different submodalities. And just note that the protein peptide fusions or conjugated or pegylated peptides are not included in the survey. It's not in the scope. And the working group received the survey responses from total 12 companies, and they are all large pharmas with more than 10,000 employees. They are actively working on the discovery and the development of the peptide drugs. I also like to mention about that it should be noted, like I pointed in the bottom of the slide, because of the relatively small number of survey responses we received from 12 companies, so the results from the survey presented here should be interpreted with caution. However, this is, as you know, it's a very rapidly evolving field. Therefore, the practice and the regulatory guidance are expected to change. So this slide highlights the 3 different subcategories of the therapeutic peptides. As described on the left side, the TPEP-A, which is linear peptides composed of only natural amino acid, we call NAAs. So platinidide is a good example with 16 natural amino acids. And the structure is shown below. And the second category is TPEP-B, which is the linear peptides containing non-natural amino acids, NNAA, and or organic linker with lipophilic side chain. So the great example is semaglutide, which has 31 amino acid and with long side chain, as you see from this structure. And it also has a non-coded or non-natural amino acid, AIB, as part of the structural backbone. The third category is TPEP-C, which is the cyclic peptides, such as cyclosporine, and you can see the structure is cyclic. And we want to also mention that the peptides with the intramolecular disulfide bond is considered as a linear peptide, not a cyclic peptide. So the example will be actually is the platinidide on the left side. You can see the disulfide bond there. So it is still considered as the linear peptide. Another point I want to make is the non-natural amino acid. It is defined as the either modified amino acid or is a D amino acid. All right, let's move on to the survey results. And the first slide here showing the submodalities, the molecular weight, and the number of amino acids. As shown in the first diagram A, and more TPEP-B and the TPEP-C peptides are under development as compared to TPEP-A. And for B, the graph B and the C, B is for the molecular weight, and the C is for the number of amino acids. And the numbers in the parentheses represents the number of total responding companies. So this is for these slides as well as for all the future slides. We did include that numbers in every graph. And also you can see from the weight, the molecular weight as well as the amino acid. So just take TPEP-B as example, and majority of the TPEP-B have a molecular weight more than 2,500 DAR and amino acid between 25 to 40. So let's move on to the in vitro metabolism studies. So the in vitro metabolite ID studies were mostly conducted for TPEP-B and the TPEP-C, as you can see from diagram A. And there's no responder actually provide that any work has been done for the TPEP-A, which is consistent with our understanding, the TPEP-A consisting all the natural amino acids. So really not much metabolism work is being done with that category. And the in vitro studies are typically conducted with a variety of biological matrices as shown in graph B. As you can see, the plasma, tissue, homogenate, and hepatocyte, a number of other matrices are pretty commonly utilized in those studies. And also in terms of timing of the in vitro studies, as highlighted in C, and most companies explore the metabolism during the lead to candidate stage and for the TPEP-B and TPEP-C. So moving into the in vivo metabolism studies, as you can see from the top A left, consistent with the in vitro studies, there is really limited studies being conducted for TPEP-A. And majority of the studies highlighted here is for TPEP-B and the TPEP-C. And also from the timing perspective, for TPEP-B, and most of the studies are conducted post-candidate and post-IND, or when requested by the regulatory. For TPEP-C, and the timing seems to be a little earlier, it's more on the hit to lead and the lead to candidate. So the in vivo studies we're talking about here is pretty broad. It includes all the in vivo metabolism studies, including all the non-radio-labeled samples from the preclinical PK or TK studies, samples from the first in-human studies, and all the radio-labeled preclinical and the clinical studies here. So now let's talk about the radio-labeled studies. And so this A, B, C are all from this survey we conducted. So the radio-labeled in vivo studies in animals were reported in all the subgroups as highlighted in graph A, but still TPEP-B and the C has occupied the majority of the space here and most commonly studied. And also in terms of the timing for the radio-labeled preclinical studies, and the most frequently studies were conducted in parallel with phase 1 or phase 2 clinical trials. And also looking into the preclinical species, from the survey results, the studies were conducted either with the rodent only or both rodent and the large animal species. Now we move on to the human radio-labeled studies. So the radio-labeled study in human were mostly conducted for TPEP-B and the TPEP-C. I'd like to point out that there was one response from the TPEP-A was in the phase 2 clinical study. So we believe that this might be the outlier of the survey. And so whatever happened in the survey results, and this is the data we gather. But based on the previous slide, we already talked about it. really there's limited metabolism work being done with TPEP-A from all the preclinical space. So this survey results, this one incidence will not impact our discussion and the recommendation in the future slides and for our recommendation purposes. Let's focus on the TPEP-B and the TPEP-C for the human studies. So most of the studies are conducted in parallel with in phase 2 and 3 clinical trials. And some companies actually conduct the study based on the request from the regulators. As you can see, the 2 TPEP-B and the 2 TPEP-C were requested by the regulatory to run the human C14 study or human radiolabeled studies. And there we do have another category for other stage. As you can see, also there are 2 responses from TPEP-B and the 2 responses from TPEP-C. And the details in the text comments there for those 2, for TPEP-B, one response is they intended to waive the study. The other response is no experience yet. And for TPEP-C, also 2 responders. One is no experience yet. The other is may apply the small molecule approach. And so if circulating metabolizes small molecule like. So this is a kind of high level summary for the human radiolabeled studies. So additional data from the survey may be continue to be discussed in the future slides. So for majority of the part, we're ending the survey results here. Let's move on to the submission packages. Those peptides approved drugs from 2011 to 2022. Not block the view. So totally we have 31 TPEP drugs approved in this period. And the peptide categories, as we highlight here, is TPEP-A, B, and C. So the definition and the typical structure of those has been described earlier. Basically, the TPEP-A is the linear peptide with the natural amino acid only, and TPEP-B is the linear peptide with non-natural amino acid, as well as the side chain with the lipophilic component. And the TPEP-C is the cyclic peptides, just to refresh the definition. So move on into the radiolabeled ADME studies in those approved drugs. So among the 31 drugs, there are 8 TPEP-As, 15 TPEP-Bs, and 8 TPEP-Cs, as highlighted in the graph A. In those 31 drugs, about 70% were evaluated in vivo radiolabeled ADME studies, as you can see from graph B here. And on the graph C here, you can see 3 different types of metabolism studies: in vitro metabolite ID, preclinical and radiolabeled ADME studies, and the human radiolabeled ADME studies. So as you can see, TPEP-B and the TPEP-C have a high percentage of conducting all 3 types of metabolism and excretion studies. And also, as you noted, the human studies were not conducted for any of the TPEP-A submissions. As you can see, the green bar, which is representing the TPEP-A, is not in the human radiolabeled ADME studies here. So this is consistent with all the, the survey results we have gained. So how we're going to handle the TPEP-A seems like the very limited metabolism work will be needed for that category. So in terms of radiolabels used in T-peptides, T-peps, different 3 T-peps. So graph A is the distribution of the labels for all those 31 approved drugs. 14 drugs were labeled with C14, 3 drugs were labeled with the tritium, and the 4 drugs were labeled with both C14 as well as tritium separately. There was only 1 drug was labeled with iodine. And the graph B is still from the survey results, and that is consistent that you can see majority of the radio labels using the C14 or tritium for all those 3 different categories. So talking about the radiolabel, using C14 or tritium has been mostly commonly used for all the radiolabeled studies from both approved drugs as well as from the survey. So then in the graph C and the D, these are also from the survey results. And in terms of the position of the radiolabel, as you can see, the majority of the cases using the label placed in the non-natural amino acid or in the organic linker with the fatty acid. And also, how many radiolabels do you typically introduce? As you can see, majority of the responses indicating the single labels for the study, and very rare has the multiple labels. So let's talk about the regulatory interaction. And in our survey, we have designed the questionnaires in such a way that there are a few questions we can gather the information about this regulatory interaction experiences. So 3 questions were asked. And the first, is has the regulatory authority requested your company to run the C14 or the radiolabeled studies? The answers were yes, and for total 3 responders, and one is regarding the non-natural amino acid. The other 2 is related to the organic linker. So 3 companies were running the radiolabel study because of the request from the regulatory. And so there's no request received for TPEP-A and the TPEP-C. In terms of the regulatory interaction about the paper argument, so the second question is highlighted in B, and so you can see how many companies submit a paper argument to the regulatory to postpone or move away without doing the radiolabeled studies. Actually, there are 3 responses from TPEP-B and 1 response from TPEP-C. They have made the paper argument. In terms of success for paper argument, we received the 1 response that was successful. They are indicating they were able to waive the radiolabeled ADME studies with published and internal data of the peptides with similar side chain information from other drugs. So there are successful cases here, depends on different category or different data package you have. So we will talk about it more in our recommendations. So let's move on to the gaps in the regulatory guidance for TPEPs. And first of all, there's no regulatory guidance on conducting the metabolism excretion studies with the TPEPs. And as you all know from the regulatory FDA guidance issued on 2022 regarding the human mass balance studies, basically the guidance indicating that the human mass balance study is not needed for drugs such as monoclonal antibody, endogenous substances, and analogs that include the peptides. Those are with no metabolism and elimination pathways based on the basic pharmacology and the non-clinical ADME information. So we define that no metabolism and elimination pathway is probably more applied to those natural amino acids, just like a protein. And so for the chemical modified TPEPs, such as TPEP-B, which contains novel non-natural amino acids and or linkers with the fatty acid, those will not really work. The metabolism fate may not be very well understood for those xenobiotic component of the molecule. So a human metabolism study may be considered. Given the lack of the regulatory guidance, as well as the inconsistency across different companies doing different ways, so it is critical to develop an industry consensus on how we're going to generate the scientifically relevant metabolism excretion data to support the development of the TPEPs. All right, so I'm going to turn this over to Sean for the second half of the presentation, which will be more related to the missed considerations as well as our recommendation for conducting the metabolism excretion studies. So Sean.
All right, thank you, Michelle. I just got the control. Oh, let me use the pointer. All right, so next I'm going to focus on the recommendations for MIST compliance for therapeutic peptide and also on conducting metabolism and excretion studies for regulatory filing of therapeutic peptide. Before I touch base on the recommendation, I'd like to make a brief introduction of the mechanism of metabolism and the excretion for peptides. So peptide metabolism is also called catabolism, is mainly mediated by peptidase to hydrolyze the amide or peptide bond into carboxylic acid and amine. The excretion of the peptide is mainly through the kidney glomerulus filtration. So we know that glomerular filtration has a molecular weight cutoff, like 50 or 60 kilodaltons. The peptide we worked on here is up to 50 amino acid. So the molecular weight is up to several thousand daltons. Definitely, they can be filtered out by glomerulus. After the peptide is filtered out, enter the proximal tubule. There are typically two main fate for them. So one is the uptaken via the endocytosis by the proximal tubular cells, and eventually digested in the lysosome into this amino acid, and then transferred into the circulation system for recycling. And the other fate is the peptide itself is digested by the brush border membrane enzymes outside of these tubular cells. Then the degraded product is reabsorbed and eventually also goes back to the circulation system. So you can see, in essence, the excretion of the peptide is also a metabolism process. That is very different from the small molecules, because the small molecule, you can either filtered out by the glomerulus or active secreted from the tubule itself. So next, I'm going to talk about the recommendation for the MIST guidance for therapeutic peptides. Do we need to follow it? In the 2020 FDA MIST guidance, it states that this guidance applies to small molecule non-biological drug products. Is a peptide considered a small molecule non-biological drug product? The answer is yes and no. That depends on the submodality of the peptide itself. And I'm going to touch base this later. Additionally, in the industry survey, most of the surveyed companies follow the MIST guidance to work on the therapeutic peptides. Unfortunately, the exact reason were unknown, maybe because they want to proactively to satisfy the regulators to follow the MIST guidance for small molecules, or maybe they really have some safety concerns for the metabolites from therapeutic peptides. But the working group think probably the first one is the main reason. However, we think whether to follow the MIST guidance should be science-based. Definitely, we should not use other modalities guidance to apply to a new modality, because peptide is a different modality from small molecule. Okay, next I'm going to briefly introduce the MIST guidance or metabolite in safety testing guidance from FDA. I think most of the audience may be very familiar with this guidance, but just for someone who just entered this industry, I want to make a brief introduction here. So MIST guidance mainly focus on the disproportional human circulating metabolites, all right, particularly the major one. which accounted for more than 10% of total drug-related material or exposure. So if there is a major metabolite, it has to be covered in exposure by preclinical safety species. Otherwise, additional non-clinical safety testing is warranted for this metabolite. If metabolite is less than 10% of total drug-related exposure, then no further testing is needed. So you can see the key message here is we need to first find a metabolite which accounted for more than 10% of total drug-related exposure. So do we need to follow, or does industry need to follow the MIST guidance? There are three scenarios corresponding to three submodalities of therapeutic peptide. The first one is therapeutic peptide containing only natural amino acid, like the therapeutic peptide A that Michelle mentioned earlier. And the second is a therapeutic peptide containing xenobiotic structure elements, like the therapeutic peptide B. And the last one, small molecule like therapeutic peptide. So I'm going to go through them one by one. So the first one, the peptide containing only natural amino acid, our recommendation is no need to follow the MIST guidance. That is because another guidance, ICH S6R1, is applied to this submodality. So this guidance is for the preclinical safety evaluation of biotechnology-derived pharmaceuticals. The scope of these biotechnology-derived pharmaceuticals include the proteins and the peptide, and also even include the chemically synthesized peptide. Apparently, the peptide containing only natural amino acid belong to the biotechnology-derived pharmaceuticals. So in this guidance, in the metabolism section, it states that the expected consequence of metabolism of biotechnology-derived pharmaceuticals is the degradation to small peptide and individual amino acid. Therefore, the metabolic pathways are generally understood. Classic biotransformation studies as performed for pharmaceuticals are not needed. So you can see for this modality, we don't need to run the biotransformation study, which means you don't need to characterize metabolites because there's no concern there. So we don't need to follow the MIST guidance. The second scenario, the therapeutic peptide containing xenobiotic structure elements. Our recommendation is may not need to follow the MIST guidance. So we know that a peptide is a linear structure, even it's cyclic, but if you chop down the peptide, eventually it's still linear, and is mainly metabolized by hydrolysis by ubiquitous peptidase. It can generate numerous of metabolites. For example, this is a 13-mer peptide, which contains a AIB non-natural amino acid. And in theory, this peptide can be digested by exopeptidase and endopeptidase at a different position to form a lot of peptide metabolites. And those metabolites can further to be digested by the peptidase to form even smaller peptide and eventually single amino acid. Of course, probably the amide bond next to AIB may be more resistant to the digestion, but the rest of part should be easily to be digested eventually. Is it possible to measure the total drug-related materials or total drug-related exposure for this peptide? Is likely no. Because unlike the small molecule, we can use radio-labeled compound to monitor or quantify all the drug-related materials. But a peptide is very challenging to use a radio-labeled strategy because it's a linear structure, right? If you label the left part, you may miss the information on the right part in the radio profile, vice versa. So because of that, you probably also think you can label each of these amino acid in the peptide, but that is impractical because there are two reasons. One, you may have stability issue if you label all of them, and the second is you may not be able to get the data analyzed because the metabolites from this, if all label the peptide, you may have metabolites containing 1 label, 2 labels, 3 labels, 4 labels. When you process data, you have to treat them differently. So because of this reason, we may not be able to measure the total drug-related exposure for some peptide, particularly longer one. And remember, for the misguidance, we have to first to check the metabolites which accounted for more than 10% of total drug-related exposure. If we cannot quantify them, then we definitely cannot define which one is the metabolite of interest. So that's why we may not need to follow the misguidance for this modality. And the last one is small molecule like therapeutic peptide. And our recommendation is to follow the misguidance because this modality is not biotechnology derived pharmaceuticals. So ICH-S6R1 doesn't apply to this submodality. And here are examples for 3 of the small molecule like therapeutic peptides. But all of them contains the repeated amide bond. So if you ask me whether they are peptides, definitely I say yes. But if you ask me whether they are small molecules, I can say no, because even they have peptide bond or maybe amide bond, a lot of small molecule drugs actually contain amide bond. And also you can see each of these units, they look like small molecules, right? The structure itself. So the metabolites from those small molecule like peptide may have some tox concern. So that's why we'll treat them as the small molecule. We recommend to follow the misguidance for this modality. So next, I'm going to switch gear to the industry recommendation or working groups recommendation on conducting metabolism and excretion studies for regulatory filing of therapeutic peptides. There are also 3 scenarios. The first one is the same as the MISA compliance, the therapeutic peptide containing only natural amino acid. And the second one, therapeutic peptide containing non-natural amino acid and or organic linker moiety, which has been incorporated in an approved drug. And the third one is the same as the second one. However, this non-xenobiotic moiety has not been incorporated in any approved drug. Okay, the first scenario, therapeutic peptide containing only natural amino acid. So this is the same as the MIST guidance. Our recommendation is no in vitro or in vivo metabolism and excretion studies are warranted. This is for the same reason because ICH-S6R1 can apply to this submodality and the classic biotransformation studies are not needed. So this is a parent suggestion. And the second scenario, therapeutic peptide containing non-natural amino acid and or organic linker moiety, which has been incorporated in an approved drug. And our recommendation, generally, no need to conduct radio-labeled ADME studies. This non-natural amino acid or organic moiety typically is a concern, right? However, if this one is already incorporated in another approved drugs, then their safety is demonstrated by the other drug in humans. Therefore, its elimination pathway may not need to be re-evaluated. For example, for the AIB, still the AIB, this seems like a popular non-natural amino acid for the therapeutic peptide. It has been incorporated in many approved drugs. For example, abaloparatide methylmerlin, and semaglutide. All those 3 drugs has AIB, and there is no safety evidence related to this amino acid. So we think there's no need to understand the metabolic fate for this AIB again. Therefore, the radiolabeled ADME study is not warranted if a peptide containing AIB. There is an exception though. So when the therapeutic peptide has a high proportion of non-natural amino acid, we may need to run radiolabeled ADME study to understand the elimination pathway of this kind of peptide. That is because if a peptide contains high proportion of non-natural amino acid, then this peptide may be resistant to the proteolysis. For example, itacacetate, which is composed of 7 D amino acid on its backbone, and this backbone was very inert to the protease. In the rat and human ADME studies, all the metabolites from this drug retained the intact backbone. So because of that, the unhydrolyzed non-natural amino acid moiety may become a novel xenobiotic structure which has never been evaluated in human previously, right? Because of that, we think we needed to understand the elimination pathway and the mass balance of this moiety. That's why for this exception, we recommended to run the radiolabeled ADME studies. The third scenario, therapeutic peptide containing non-natural amino acid and/or organic linker moiety, which has not been incorporated in any approved drugs. Our recommendation is, in general, to conduct radiolabeled ADME studies, because this non-natural amino acid or organic linker moiety has never been proved to be safe in humans by a drug, then because it's also a xenobiotic moiety, so we think we should run the radiolabeled ADME study to understand the elimination pathway and the metabolic fate of this moiety. There is also exception though. So for example, if a peptide incorporated this non-natural amino acid or organic linker, but we have sufficient data or literary data to demonstrate the safety of this moiety, then radiolabeled ADME study may not be needed after the endorsement from the regulatory authorities. Okay, so this is the decision tree for the industry recommendation. This is a decision tree actually recap what I just mentioned. Briefly, first, we need to check whether the therapeutic peptide containing any non-natural amino acid and/or organic linker moiety. If it is no, then no metabolism or excretion studies are warranted. If it's yes, then we can check whether the non-natural amino acid and/or organic linker moiety was incorporated in approved drugs. If it's yes, then no radiolabeled ADME studies are warranted. If it's no, then we can also check whether we have additional data or literature to support the safety of this xenobiotic moiety. If we don't have any, then the radiolabeled ADME study may be warranted. If we have, then we can consult with the regulatory authorities. If they also agree, then we don't need to run the radiolabeled ADME studies. Otherwise, radiolabeled ADME studies is recommended. Okay, this is my last slide before the acknowledgement slide. We presented the industry survey results to demonstrate the current industry practice on the metabolism and excretion of therapeutic peptide. We also presented the result from the NDA and MAA submission documents for the therapeutic peptide. And those results provide some very valuable information on regulatory expectations for metabolism and excretion of therapeutic peptides. We also provided the recommendations on the MIST compliance for therapeutic peptides and on the conducting peptide metabolism and excretion studies for regulatory filing of therapeutic peptides. Lastly, we believe that the best practice for therapeutic peptide metabolism and excretion will be established with emerging data and state-of-the-art technologies and continuing efforts between industry and the regulators. So this presentation is based on the work from the IQ Peptide ADME working group. So this is actually a combined effort from all the members. We thank to all the members. They are brilliant scientists. We are pleased to work together. We also like to thank IQ sponsors, Jasper and Wenqi, and the early members for this working group. We thank IQ Secretariat, IQ Translational and ADME Sciences Leadership Group, and the IQ member companies which participated in the industry survey. Lastly, we'd like to thank IQ. Without this organization, there is no presentation today. Okay, this is the last of the last. The content of this presentation was just published at the Drug Metabolism and Disposition Journal. This published white paper contains more detailed information than what we have presented today. If you are interested, we highly recommend you to read this paper for more useful information. Thank you for your attention. We're happy to take any questions. Jasper?
Thank you, Sean. I was a little bit worried that it was all too crystal clear because it took a while before the questions start coming in, but now we do have some. I think we will start with an easy one, which others are probably thinking about regarding slides. And yes, anybody who signed up will receive a recorded link and the slides for this webinar. So the first question that came in is from Grosset Campbell. For the ADME studies, why is there a preference for using radioisotopes, for example, carbon-14, compared to stable labeled isotope carbon-13? So I just put the question out there, and Michelle, Sean, and Constance, anybody want to grab this one?
So yeah, maybe we can address this question. For the ADME study, is include the mass balance study. Okay, for the mass balance study, which means you need to quantify the drug-latent materials in different matrix, such as urine and the feces or bile for the animals. If you use C13, which is a stable isotope, so this one, there is no good way to measure them, all right? For the C14, why we use that? Because there's a radioactive detector, and this one can detect, this C14 is ideal for the quantitation of all drug-latent materials. In this case, then you can calculate the mass balance for the excretion in the urine, feces. Additionally, when you run the radio profile, then you can quantify each of metabolites in this radio profile. If you use the stable isotope C14, you are not able to do that. That's why we use C14 or tritium, any of the radio. Most of them we use C14 or tritium in the pharmaceutical industry for this purpose.
Thank you. Anybody else has anything to add?
Very well said.
Sorry, Michelle, I didn't get that.
Yeah, it is, the reason has been stated clearly.
Okay, good. We move on to the next one from Gian. So based on the survey, it looks like there is a higher rate for ADME studies, non-radio-labeled or radio-labeled for TPEP-B. Is that mainly because they contain non-natural amino acids?
Michelle, you want to go first? Yes, yeah, I think so. That's where the focus is on when you have a xenobiotic component of it, meaning either non-natural amino acids or the linker, the fatty acid. Those are more kind of small molecule-like component of the molecule. That's where that triggers more studies to characterize the fate of the metabolism. I think the regulatory probably perspective as well, they want to understand what's going on with those non-natural components.
Yeah, I agree with that. Definitely, that is because the therapeutic peptide B, we define that which contains either non-natural amino acid and or lipid side chain, which both of them are xenobiotic moiety, right? If you have this xenobiotic moiety, then you may have some concern that you want to know how this moiety is excreted from animals and humans compared to the T-peptide A, which is composed of only natural amino acid. Hopefully this addressed Dan's question.
Yeah, and please for the people out there, if you don't think your question was answered to the level you preferred, then make a comment and we might take it up again. We move on to the next one. So how about the case of TPEP-A with unnatural amide binding? Anybody up for that one?
Unnatural amide binding. So I'm not quite sure what does that mean. If this doesn't mean that the amide bond is modified, then that is not a T-peptide A anymore.
Yeah, I was thinking the same thing.
Yeah, because if you modify the amide bond, which means this is amide bond itself is non-natural, and we should classify this as a peptide containing xenobiotic moiety. But I really don't understand what do you mean binding mean.
Yeah,
unnatural amide binding.
I think we addressed it. Let's see if the person can add more if we misunderstood the question.
Okay.
We go on to Jan Sandel, who has two questions. We start with the first one here. Are there any in vitro metabolism approach which has been shown to be predictive of the in vivo metabolism? That's a good one. Do you want to? Yeah, you want to go, Constance?
So the blunt, general, high-level answer is there is no general approach or one size fits all. However, in some kind of subgroups of peptides or some submodality, subspace, you can find relationships between what you observe in plasma or what you observe in hepatocytes for some within a project. But unfortunately, cross-pharma and with the different modalities that we have looked at and the different assays that we have looked at and compared, there is no beautiful, use the liver and use plasma and you will be home by using a scaling approach.
Yeah, I mean, you guys can also add comments here. Definitely, peptide metabolism is very different from small molecules because the universal presence of peptide days in the body, right? So you cannot use a small molecule approach to process peptide. Like a small molecule, we know the major metabolism enzyme, so it's much easier to predict in vivo. But peptide depends on the purpose of the peptide, even the delivery route of peptide. Then you may want to use a different matrix for the in vitro to predict in vivo. I think we have relatively detailed information in our white paper. There is table 2. You definitely can look at this.
Good, thank you. We'll move to the next one from Jan. Oh, did anybody have anything to add? I'll continue. So is there any scenario where a non-clinical ADME study in primates could be considered as necessary, as it could be considered as ethically questionable? That's the question. Anybody want to start on that one?
You guys want to go first? I mean, I can address. I can provide my opinion on that.
Let's start with you, Sean.
Okay. So I think this is, I mean, it's a good question. I don't think it's necessary to run non-clinical ADME in primates for therapeutic peptide. Actually, even for the small molecules, there's also debate whether the preclinical ADME study is needed or not, right? So some company only run one just in the rodent. I think it's the same as the peptide. We don't see there's a special reason for the peptide. We need to run the ADME in non-human primates.
Yeah. Anybody want to follow up?
Yeah, I would agree. It depends, right? How much understanding you have with the peptide. And the rat study, certainly it will be needed, especially if you plan to run the human study. You need to have a QWBA and other rat study to calculate the dosimetry. So in terms of the large animal species, and if you have a data in general is consistent with the other species in rat, and that study, may not be critical and can be done, maybe postponed to later time. Those are another approach can be utilized, may not be necessary in parallel with the rat study. So, but again, it depends on the nature of the peptide. And also Sean mentioned about the proportion of the non-natural amino acid. If it really behaves like a small molecule, you probably want to do both. And so this is just general consideration.
Yeah, additionally, I think there is no clear evidence to show that the non-primate protease is more similar to human protease, I mean, in terms of metabolism. So it seems like there's no species difference between rodent and non-rodent for the peptide metabolism. No clear evidence to show that. If you say, okay, non-primate may have a very similar metabolism pathway for the peptide to the humans, then maybe we should use non-human primate. But it seems like there's no literary data to show that so far. So we think rodent may be enough.
Another point to consider, I want to point that is the large animal species depends on the therapeutic area. If if the large animal species is more kind of a target related, active in pharmacology. So if you really need to focus on that, so the select species, which is relevant to human, right? So if from that perspective, that will help you select the species for the radio label study.
Yeah, as you mentioned that, I mean, if the monkey is a tox species, and let's say that you actually see some tox there, which you need to address, could be caused potentially, or have to exclude be caused by metabolites. I mean, that could be a reason for looking into it.
That's correct.
Yeah, definitely.
Whether you need a radio label, that can always be discussed. But for mass balance reasons, perhaps you want to create a larger package to try to avoid the human ADME study. I mean, that is sometimes successful. But of course, it has to be considered for ethical reasons. Constance, do you have anything to add to this one?
I think we have discussed it in many dimensions, in many directions. But to summarize it into a nutshell, it is that a non-human primate ADME study for a peptide would be an extremely rare case. It was nearly like it has to qualify like a small molecule, which were a non-human primate ADME study is warranted. Doing monkey studies within your development program, of course, having extra plasma or collecting urine, we may look for metabolites there, as we have those samples, but to design an ADME study, it would be a brain stretch today to come up with a scenario where it's really warranted.
Yeah, also keep in mind that things develop, right? I mean, what we do today and what we did 5-10 years ago, there's probably a lot of differences.
Yep.
So we take the next one, Faraz Kasmi. Oh, I did not pronounce that right. Sorry about that. Based on the survey result, was there any discussion within the working group on best in vitro test systems to evaluate peptide metabolism? So that touches upon the one we had before. So Constant, can you briefly take that one again?
Because there was a lot of discussion which would be the best system, but we could not identify the one single best system. That would be the short answer. Maybe in the space of small molecule Pepsi types, those which are so small molecule like, well, we fall back to hepatocytes and cytochromes, but when we have peptidases, there is unfortunately no oh, take this in vitro from the shelf and you will be home.
Yeah, I think just check the table 2 in our published white paper. There's some information there.
Yeah, so a lot
Yes.
can be used, but it has to be kind of tried on a project basis, what does work or not.
Yeah, it really depends on the delivery. You know, if we just have a sub-Q, IV kind of dosing circulating, or if you have developing the oral peptides. So that's where the in vitro system might be different. If you look at the oral delivery, so certainly, you know, the GI fluid or those assimilated fluid may be relevant to look into that. So it, you know, yeah, the table we highlight is really for different purpose or different delivery. And so that gives you more idea for what in vitro system might be relevant.
Okay, I think we move on to the next one. Beverly Knight, do the recommendations for human ADME studies change depending on the route of administration? For example, oral peptides. Anybody want to take that one?
I don't think so, because this same as the small molecule, right? If you IV or oral, small molecule, you needed to do the same, typically same route of the ADME study. So the same as the peptide.
I guess the only thing that could change is if you want to do a radio-labeled human, I guess that's the question here, label study with an oral run. I guess you should do a bit of calculations if you at all would be able to measure it, because depending on the bioavailability, that might be a challenge. Yeah, that is, of course, that's the exact case. So even in the mass balance draft guidance from FDA, is mentioned that if there is no or minimum probably like a circulation of the drug-latent material, then you probably don't need to run the human animal study, which means if your oral peptide only have 0.1% oral bioavailability, maybe you don't need to run. So that's it. I think this is also debatable. But if you have some absorption, for example, 5%, 10%, definitely you need to run the human mass balance if it's needed, right? 5-10%, that's not bad, right?
Not bad, because it's
You
in the
could get that.
circulation. It's in the circulation,
Yeah.
then you should do the mass balance to evaluate it.
Yeah. Any further comments to this one? No, we'll go further. So is the natural abundance of carbon 13 much higher than carbon 14, which will interfere with quantification? So
That's about what Sean was saying.
Sorry?
That is about what Sean was saying.
Yeah.
15 minutes ago.
So I think we have make some noise if you don't think we addressed this one properly.
I think this is a good question. Actually, for the quantitation of C14 is not depends on LCMS. All right. So we're not seeing the LCMS to quantify them. Is using radioactive detector or AMS. So you for the C12, C14, C12, C13, it will not be measurable by those detectors. So they are not going to interfere with C14 detection at all.
Yeah. We move on. Raymond Evers, based on your experience, are there any examples of macrocyclic peptides that show hepatic excretion? For instance, a peptide like CCK8 is a substrate for OATB 1B3.
I could take a go at that. That's a very good question. Looking at the structures, it is a natural peptide. A macrocycle is a cyclic peptide. And with that cyclization, the macrocycle is kind of disguising itself peptide properties and sometimes rolling over into what we observe or perceive. Also, what is small molecule. So cyclosporine maybe is even more extreme than CCK8.
Constance, you broke up. Could you just repeat the last 10 seconds?
Oh, I'm sorry. Now I need to remember what the last 10 seconds. But so where I was going is that the peptide, which is cyclic, is more kind of taking the structure and the shape of a small molecule, particularly as those have maybe 8, 9, 10 amino acids. So we're not talking about the ones with 30 or even more. And those actually may have membrane permeation as small molecules. They may be recognized by transporters. They may be recognized by the metabolizing enzymes that we know from classical chemicals. And there we also find ourselves, which is not in scope of this work, but for thinking ADME, where we also may find them being subject or causing DDIs. So there actually we are in this peptide TPEP-C space or small molecule-like space and have to tune those eyes. There's not much experience in many examples, but looking at the structures that we have been looking at from the submission packages, this is where the story goes. So yeah, Sean's example, is it a peptide or is it a small molecule? It may be both.
That's a long discussion.
Yeah. I think I never work on the microcyclic peptide, but we know that cyclization of peptide can enhance the permeability. And also some of cyclic peptide has a small molecule property, right? They are the substrate of some of the CYP enzymes. So it's potentially, if we have a lot of cyclic peptide developed in the future, potentially you may observe that maybe excreted from bile is possible. I think it's possible.
Yeah. Can we close it here or further comments on this one?
Go on, we have a lot of questions.
Sorry, Constant, what did you say?
We have a lot of cool questions waiting, so let's go on.
We do. So the next one is from John Griffin from Zealand Pharma. We have a case with 2 active metabolite peptides with above 10% of total for both, all natural amino acids. We are following PK exposure for all of them. If no radioactive /cold med ID is performed, then how do you follow them?
So, okay, do you guys want to go first?
Go ahead, Sean.
Okay. So I think this one is a question is how to measure those metabolites quantitatively, probably using radioactive, right? Because you need the med ID. So our recommendation in the presentation of white paper is actually recommend for conducting metabolism and excretion studies for regulatory finding itself, okay? We definitely don't include any other studies which may help your teams. For example, like a metabolic soft spot in the early stage, you may want to determine what is soft spot of your peptide, but this information is not needed for the regular finding. But for you guys here, this is something special. You need to identify those active metabolites, right? You want to quantify them. Yeah, you definitely can use whatever way you want to do. You can use radio labeled or cold med ID. I don't think we're against that. You definitely can do that. So it's just this one will help your team to understand molecule more, but this piece of information may not be needed by the regulatory finding for your therapeutic peptide. I hope this will address your question.
Yeah, in that case, I would think instead of doing the radio label, if you know the 2 metabolites at the high percentage, say more than 10%, so you can simply just develop the bioanalytical method, right? Just to see what's the quantitative perspective, what's the the concentration of it. That could guide SAR, actually, especially in the discovery. And if you think the metabolite is higher a certain degree, and the bioanalytical method will be the easy way to monitor it quantitatively.
Mm-hmm.
Yeah. Additionally here, percent of total of both, I think this is probably not a percent. The total drug-related material may not be accurately measured here. So probably it's an estimation. You may find the 2 metabolites and the parent and maybe another metabolite, then you calculate total, but you may also miss the others.
Mm-hmm.
That's my guess.
Sorry.
Yeah. Sorry.
I was about to give a different tweak to that answer because we have now been very, very technical, but maybe Joe's question was more teasing out after this is only natural amino acids.
Yeah.
But still, our recommendation would hold as now here, these metabolites are active. So there is an additional purpose to understand their exposure, as Ms. Michelle was saying, maybe just having a BA method to follow them, because there's another trigger and another reason to quantify and to characterize their fate and going around. So it's not this only natural amino acids. There may be, as Joe is having this nice case, other reasons to and still do follow-ups and perhaps even labeled.
Agreed. Yeah.
I hope I understood it correctly what he was out after.
Yeah, and as for the other questions, if you didn't think we addressed them correctly, just put in another comment and we'll revert to it if there's time. But we have 15 minutes left, so let's see how it goes. The next one, Manu, have you observed a scenario where in a case the T-PEP conjugated with a small molecule, the partial metabolism of linker and amino acid could result in alterations in metabolism such as reabsorption due to amino acid transporters? Do you want the question again?
So we did state in the scope earlier in terms of conjugation with other components. It's not in the scope of our working group discussion, I guess. I don't know if this is falling into that category.
Yeah,
But do we have any examples
I think
of that?
no. I think this
Do we
is
have
a
example?
very
It's a
special
good question, really. Special
Uh,
question.
But this is a little different from what we say because this is a small molecule peptide, so it's still very small. We can call it a small molecule like peptide, maybe, right? So, this question is very interesting. I think definitely if the peptide linker is cleaved, then the metabolism of this moiety can be changed, just like a small molecule, right? If you have hydroxylation or you chop out this one part of molecule, then metabolism will be changed because the enzyme will recognize it as a different structure. But in terms of reabsorption due to the amino acid transporters,
There, I think we can make a point because amino acid transporters are there to transport amino acid, which is small amino acids, or there are different types of amino acid transporters for different substructures, whether they are charged, and how they are charged, and how large or lipophilic they are. But those would not accept tripeptides or tetrapeptides or small molecules of larger size as their substrate. So they're really, really there for making those tiny molecules go across membranes. So I would not assume that if I'm having a small molecule with one peptide left on the side, that would become a substrate. We don't have to be afraid of that situation.
yeah.
Good.
I think the simple answer to this one is no, we have not observed that. At least I have not, because I never work on the peptide small molecule conjugate. That is a very special modality.
Okay, we move to the next one. So Brad Maxwell, why were pegylated peptide excluded from the survey? Are there plans to include them in a future survey?
Oh, so so I think we discussed this, right? Initially, the pegylated peptide is very different because it's considered a large molecule, definitely large molecule because it have a much larger molecular weight, and also we are more focused on the peptide itself instead of the pegylate, because peg is different, right? So peptide itself, which means we have these three subcategory, and if you pack these three subcategory, then you can use the recommendation for these three category to treat with the pegylated peptide. I think eventually you probably will do that, because if you pegylate a peptide which contains the zero biotic moiety, I believe you need to understand the elimination pathway of this peptide itself.
Yeah.
Do you think so?
Yeah, and just the last part there, I mean, there are no plans to include this in any future surveys at the moment, right?
No, I don't think so.
Okay, we go to Marcus Wallace from Novartis. You explained your rationale for not performing ADME studies based on safety. If you have clipping of peptides leading to other active peptide species and there are species differences, would you not consider additional profiling?
If the metabolites are active, we need to understand how they are active. So that's then opening another chapter in that book. So Michael is right with that challenge. Then we have another challenge.
Yeah, still, I think this is also still very special, right? If you have this active metabolites and you want to investigate further the mechanism, then yeah, you can do the additional profiling. So we are not recommend to not run any of the like a metabolism study. What we recommend is only for the regular finding, right? Because regular finding typically they only focus on the safety of the drug itself. But if you have other reason to run the metabolite profiling, then do it. Just the same case like this, I think you can do for sure. And the metabolite profiling is a very useful tool for other purpose for sure.
Yeah, and also as you can see in our decision tree, even we say, if you have a amino acid, non-natural amino acid, which has improved the drugs. So although the C14 radiolabel study is not warranted, but we still are recommending, doing non-radiolabel study. You're still welcome to do more studies if needed. That's where in this case, if you have active metabolites, you have something special, you do have some concerns, or you expect some concerns. We certainly encourage you to run the study or doing some, non-radiolabel study to see where you're at, if you really need to do the radiolabel study or not. So there are some gray areas. Like I said, we are not saying you should not run it. We just say in certain cases, in general, it's not warranted, but there are many cases you probably do need to run the study.
Yeah, I think we have a general recommendation. Definitely there is some of the exception, like what I showed in the presentation too. Just like a human mass balance draft guidance, they also have exception. All this guidance is general. But thank you. Good question.
Yeah, we go to another good one. Stephen Lang, did you consider local peptide delivery, for example, ocular, with low systemic exposure? How would this change the decision tree?
Yeah, that's a really good question. We definitely discussed this previously, right? Is local, you don't need to, just like small molecule, you don't need to run the radiolabeled ADME study, right? If you use local, even for the small molecule. So my opinion is we don't need to run the radiolabeled ADME study, no matter it contains xenobiotic moiety or not.
No, shouldn't be necessary.
No, because there's no
systemic
exposure. Yeah, this is also one exception in the human mass balance draft guidance issued by FDA last year.
Okay. We move on to the next one. So, great talk. Question to Sean. There is a 10% threshold for metabolites to trigger the identification process. What if metabolites below 10% threshold are way more toxic than the intact and there are tox concerns? How should we specify that?
Yeah, this is a good question. So for the MIST guidance itself, is it still a general guidance focused on the more than 10% the total drug-related materials? But they also say, there are some special case. If there is a metabolite which is toxic, no matter it is 1%, 2%, or 5%, if it is toxic, for example, in the animal species, then we definitely cannot follow this misguidance. Just say we needed to characterize the metabolites with more than 10%. The toxic one is exception, right? If you see this metabolite in the animal is toxic, then in the human being, even is 1%, I'm not sure is acceptable level or not. So definitely is the special case, then we needed to characterize this more carefully. Then the general guidance may not apply.
Yeah.
Yeah.
Move on to the next one. Oh,
Good
anything?
answer.
Okay, thank you. We move on. So we have John Griffin back on the stage. Do you think that a non-clinical ADME study would be sufficient when the non-natural amino acid is similar, but not exactly the same as previously approved non-natural amino acid? Show excretion is same as previous. So
You guys will go ahead.
Can I clarify what means similar but not the same? Just
so
different-
it must be a different one.
Minor change of the
minor
structure.
changes, modifications.
Yeah, I think in our
Yeah.
recommendation tree, we state that, right? We have this states that if there is a literature evidence or other sufficient data to show the safety of the novel moiety, then we may not need to run the radio-labeled ADME study. So the same here, this one is similar to the non-natural amino acid that is incorporated in the approved drug, but they have a slightly different structure. However, if you have some data to show these two moiety are very similar, I mean, like in terms of metabolism, and then maybe you can discuss with FDA or other regulatory authorities, and hopefully they also agree with you, then you may not need to run the radio-labeled ADME study. But you
Yeah,
guys,
that's where the
comments,
people,
right?
yeah, that's true. That's where the, the decision tree on the people argument. If you believe your molecule is so similar with the drug already approved, and that's where maybe have a conversation with the regulatory, and maybe you have some data, even non-radio-labeled study to demonstrate that, to ensure, that's the similarities that may help you to make the argument.
We'll take the next one. Sekar. In the recommendations, you state TPEPs with high proportion of non-natural amino acids may need to conduct radio-labeled ADME studies. What proportion do you consider high? Any cutoff metrics do you suggest?
This is a very tough question, a really good one too. So I don't think we have this cutoff value right now. Definitely each of the sponsor, they need to think about this, and it's always case by case. I think the risk benefit, you need to evaluate that. But we don't have any of these suggestions right now. Hopefully in the future, with more data generated, we may have some, like a 50% or something like that. But right now, I don't think we have. Constance?
To add on, it's a similar scenario as in the question before. Well, as a molecule is developed, you gather experience. That is the metabolism. In animals, you see whether maybe 5 non-natural amino acids in a row is resistant. It's there for all the time. You observe that in your first human trial, see it there as well. It will ring a bell. Okay, maybe I have to do some extra work on that one. Unfortunately, we have not, from the literature and the submission packages, have had those examples that we can tell the story, but that's what we would anticipate.
Yeah, I think a lot of cases is case by case because this peptide modality is very diversified, right? Some of them maybe 5 amino acid, non-natural amino acid, they are separated, distributed in the protein, the peptide, or all of them stick together. Then you may treat them differently.
Yeah.
Yeah.
We go to the next one. We check the next one here. Have you looked at the excretion and metabolism of larger TPEP that is over 200 amino acids? That might be what might be the issue with these larger peptides? Well, I guess we can start with our definition is that if it's above, did we say 40 or 50? I think we 50. Said 50.
Yeah.
Then we define it as a protein, so it's out of scope. And generally, you could follow a section. You don't have to do much here.
Yeah, I see. This is a protein, not a peptide. Yeah. So we should follow the guidance from the protein instead of peptide.
Yeah. Okay, we only have 1 minute left, so we won't be able to touch on all of them. I just see if we can find something. Okay, let's take this one. Any suggestions regarding radio-labeling position for the type B TPEP containing non-natural amino acids, which is recommended for radio-labeled ADME study?
Yeah, so for this one, I think we talk about this. So we prefer to label the non-natural amino acid moiety or the organic side chain because those are the xenobiotic moiety and which is the potential safety like a safety reason, right? That's why we are focused on that. For the natural amino acid we know is elimination pathway, then we definitely don't need to label them. At least it's much low priority.
I would like to add a consideration. So think non-natural amino acids, which may not be incorporated further, and having some lipid on it. Well, often we have those lipids, and those things are the ones which are protein bound, which circulate quite long. So it might be a good idea to follow the thing which is around the longest and put the label into the lipid.
Yeah, I mean, you can also label both of them, like 2 labels. Yeah. I see there's a survey answers. There are some company label them multiple label, actually, just 2 labels, like.
Yeah, I think we have to stop here. We haven't made all the questions and comments, but if you would like us to address them, then please reach out by mail. In the slides, there are email addresses for Michelle, Sean, and I, at least. We don't have your email addresses, so we can't go the other way around. And thank you for listening in, and we hope you enjoyed it.
Thank you so much.
Thank you, everyone, for great questions. Have a great day.